Welcome to Lecture 11.1, in which we will go over DNA. There have been many scientists who have contributed to our understanding of how DNA works, and here are a few of them. This is Johannes Friedrich Miescher, who was the first to isolate nucleic acids in 1869. However, several decades passed before the structure of DNA was identified. Rosalind Franklin is known for perfecting the technique that allowed for her and her student to obtain the crystal structure of DNA. Watson and Crick, depicted here, are known for discovering the structure of DNA, which they were able to do with the help of the DNA crystal structure obtained using Rosalind Franklin's techniques. Since then, many more discoveries about DNA have been made. Our learning objectives for this video are to explain the central dogma of molecular biology, describe the structure of a DNA nucleotide and distinguish between the different bases, understand the directionality of DNA, describe the structure of double-stranded DNA, and learn how to determine the DNA sequence of a strand, given the DNA sequence of the other strand. Let's begin with the central dogma of molecular biology, which is the foundation of our understanding of how genes are made into proteins. We start off with DNA that stores our genetic information inside the nucleus. The genes encoded by the DNA are what determine our genotype, which we learned about in the genetics lecture. During cell division, the DNA must be replicated to ensure that each cell has a copy of DNA. Since not all of the DNA is needed at once, when cells need to use the DNA, they will instead make a temporary copy in the form of messenger RNA, abbreviated mRNA. This is done through the process of transcription. The mRNA is then exported from the nucleus into the cytoplasm. The ribosomes in the cytoplasm, or on the rough ER, then take this mRNA and use it to create proteins in a process called translation. The proteins that are made are what ultimately determine our phenotype. In this video, we will focus on DNA. What is DNA? Recall from the macromolecules lecture that DNA stands for deoxyribonucleic acid, and that it is the genetic material found in all living organisms. Recall also that DNA monomers are called nucleotides. Nucleotides are composed of the 5-carbon sugar deoxyribose in green, phosphate groups in orange, and a nitrogenous base in blue. You will want to know that the 5-carbon sugar is numbered such that the first carbon is the one attached to the nitrogenous base, and then the numbering increases as we move to the left. The most important ones to know are that the 3rd carbon is attached to a hydroxyl group, and the 5th carbon is attached to the phosphate group. DNA has 4 different nitrogenous bases. Adenine and guanine, referred to as A and G, are purines, which are nitrogenous bases with 2 rings. Cytosine and thymine, referred to as C and T, are pyrimidines, which are nitrogenous bases with 1 ring. As we will learn, a purine will always form a pair with a pyrimidine, and specifically, an A will always pair with a T, and a G will always pair with a C. How is a strand of DNA formed? Here are 2 nucleotides. What happens is that the hydroxyl group attached to the 3rd carbon, also known as a 3' carbon, forms a bond with the phosphate group attached to the 5' carbon of another nucleotide. This creates a phosphodiester bond, shown in black. The 5' end is where the first nucleotide is, named for being the side where the 5' carbon is sticking out, and the 3' end is where the last nucleotide in the sequence is, named for where the hydroxyl group at the 3' carbon can go on to attach to another nucleotide. As a result, DNA forms a direction in which it begins at the 5' end and ends at the 3' end. While that is how a single strand of DNA is formed, DNA is almost always found in a double-stranded form. The 2 strands are held together by hydrogen bonds, depicted here as dashed lines. The 2 strands form base pairs. As we mentioned, A pairs up with T, and G pairs up with C. These base pairs are complementary, which means that if A is on one strand, T must always be directly across from it on the other strand, and vice versa, and it's the same for G and C. Notice that A and T form 2 hydrogen bonds, and G and C form 3 hydrogen bonds. This makes the bond between G and C stronger, so DNA with more GC pairs are generally more stable. Another important aspect of double-stranded DNA is that the 2 strands are antiparallel. This means that the directionality of the 2 strands is always the opposite. So the 5' end of one strand will be on the opposite side as the 5' end of the other strand, and the same for the 3' end. Overall, the two strands of DNA form a double helix. It's a bit similar to a spiral staircase. The phosphate groups create a backbone, and since phosphates are negatively charged, this gives DNA an overall negative charge. These are like the handrails of the spiral staircase. The double helix is formed by how the bases pair up through hydrogen bonds. These are like the steps of the staircase. The complementary nature of DNA means that if we know the sequence of one strand of DNA, we can easily figure out the sequence of the opposite strand. The opposite strand will always be complementary and anti-parallel relative to the original strand. For example, say we have this random sequence here, with the 5' end on the left and the 3' end on the right. Since the opposite strand is anti-parallel, we know that its 3' end is on the left and its 5' end is on the right. We can then figure out what the complementary sequence is. Let's start at the 5' end of the opposite strand, since DNA is made in the 5' to 3' direction. We have a T here, which pairs with an A, so this nucleotide must be an A. Next we have a G, which pairs with a C. We can then continue to put in the complementary nucleotide for the rest of the sequence. If we want to write the sequence out in the 5' to 3' direction, but from left to right, all we have to do is reverse the sequence. In this video, we learned about the central dogma of molecular biology, which states that DNA is used to make mRNA through transcription, and mRNA is used to make proteins through translation. A single DNA nucleotide is composed of a 5-carbon sugar, phosphate groups, and a nitrogenous base with either one ring, which are the pyrimidines C and T, or two rings, which are the purines A and G. A strand of DNA is formed by the hydroxyl group on the 3' end of one nucleotide, forming a covalent bond with the phosphate group on the 5' end of another nucleotide, and this creates a 5' to 3' directionality. The double-stranded DNA forms through hydrogen bonding between complementary base pairs, such as GC and AT, and the two strands are anti-parallel. Because of this, if we know the sequence of one strand, we can figure out what the sequence of the complementary strand is. In the next video, we will go over DNA replication and repair. Thanks for watching.